So my talk today is on trying to piece together all these different lines of evidence. I think, now, at least in the papers that have been published in the last 2-3 years or so, allows us to, I think, to get a better handle on, what really protects against dengue and what causes disease. And okay. So because I'm going to be the first of a series of 3 talks, I thought and I have a little bit more time, so I thought I'll introduce dengue to the audience. So this is a distorted map to show you the burden of dengue, and you can see that, in Southeast Asia, in Central and South America, and South Asia, that's where the dengue burden is, and it's estimated that there's about 100 million cases of dengue each year. Africa is a bit underrepresented, but there's a lot of emerging data now that there's probably a lot more dengue than what's appreciated in Africa. I think part of it is because of the way fevers are managed in Africa. Because of endemic malaria, then they're usually treated with chloroquine and diagnosis clinically as malaria. But with laboratory evidence now, it's showing that there's a lot more dengue and chikungunya in Africa. This is the genome of dengue virus. It's a positive strand RNA virus. It's a very small virus, about 11,000, slightly less than 11,000 nucleotides in length. And I always joke that anything longer than that I'm not interested in it. It's just too complicated. So 11,000, it contains 10 genes. These are the structural genes that form the virion. So it's an envelope virus, so there's a lipid membrane in which the envelope and the membrane protein sits on it, and then inside is the capsid, which then encapsulates the RNA, the positive strand RNA. I'm telling you this because it will come to it. It will be important later on. So the capsid is preformed. All these enzymes are non-structural. They form the replication complex of the virus to replicate RNA. And so they are only expressed upon infection and inside the cell. They're not packaged into the virion. Okay? So this is on the left-hand side is the phylogenetic tree of orthoflaviviruses. In the past, it was called flavivirus, but now they've changed it to call it orthoflavivirus. Dengue is up here. The silhouette are the vertebrate hosts. They're all transmitted by arthropods, either mosquitoes, sandflies, or ticks. You can't see this here very well, so I've blown it up here. And so you can see this is Zika virus is here, dengue 4, dengue 1, dengue 3, and dengue 2. The point I want to make here is this. If you look at the length of these branches and the length of these branches, they are about the same. Here we call them species, here we call them serotypes. I think they're all different species. Dengue is caused by 4 genetically different viruses. We're just stuck in this historical nomenclature that, because the viruses were named before genome sequencing was available, then we call them serotypes. So effectively, they are 4 genetically different viruses, and they behave differently. They are all transmitted by this mosquito, Aedes aegypti, principally, but other mosquitoes like Aedes albopictus can also transmit it. So the idea of why I'm telling you there are 4 different viruses, because it is thought that infection with one will give you long-lived immunity. Now, how true that is now debatable. But you now remain susceptible to the remaining 3 dengue viruses. And it's a second infection that tends to be associated with severe disease. And the notion of that came from this epidemiological observation in Bangkok. So this is the work of idea of Scott Halstead. And his story was that he used to work in Bangkok at this Armed Forces Research Institute on Medical Science, or AFRIMS, in Bangkok. And it was on a flight back from the US that he thought of this idea that in this age group, children 1 to 5-ish, they're experiencing their first infection. And the rates of severe dengue is lower than those children experiencing the second episode of infection. Right? So he then postulated that this is immune enhancement. And to support that theory, there's this group here, the infants, because of waning levels of maternal antibodies in the blood, and then there's elevated risk of severe dengue at about 6 to 9 months. Then together, he postulated that this dengue is caused by, or at least severe dengue is caused by this antibody-dependent enhancement of infection. And the model for that is explained over here in this cartoon, where because dengue virus infects myeloid-derived cells like monocytes, macrophages, and dendritic cells, which express Fc receptors, so dengue has effectively two entry points. Firstly, a canonical dengue virus receptor interaction. What exactly is a receptor is not definitive, but it's probably several receptors. There's not just one. But if an antibody binds, a non-neutralizing antibody binds dengue virus, then it can also enter myeloid-derived cells through the Fc gamma receptors, and collectively then cause this increased replication and therefore the antigenic burden that drives inflammation. That's the theory. Now let me show you the real life. So that's the in vitro part. But what happens clinically? This is a study from Vietnam, and I would encourage those of you who are interested in dengue to go through it in detail because it's really interesting. There is another paper that accompanied this. This was published in 2022 in Clinical Infectious Diseases. But this is a collective data of 4,000 over children with dengue, diagnosed with dengue, and with blood drawn daily to measure the viremia. And that's how they can reconstruct these curves. So this is, to orientate you, there's dengue 1, 2, 3, 4. In blue are primary, and in red are secondary dengue. And actually, you can see there's no difference in viremia. If anything, the dengue 1 viremia in the primary context is higher than secondary, which is completely contradictory to the way we think of antibody-dependent enhancement, which is an enhancement of virus replication in myeloid cells, but you don't see any clinical evidence of that, right? Instead, what you see is this increased risk. So to orientate you again, this is dengue 1, 2, 3, 4. This is by day of illness. In x-axis is the viremia levels measured, or rather RNA-emia, is a qPCR. And then on the y-axis is the probability of severe dengue, right? And you can see the red line is still secondary and blue lines are primary. So this, for viremia, per viremia unit, the risk of severe dengue is much higher with secondary than primary. But even though from day of illness, there's no difference in the viremia levels between primary and secondary. Yet somehow in secondary dengue, you're still getting greater risk of severe disease, suggesting that it's not just viremia, that there's some other things going on in terms of the host response to the secondary dengue infection, which I don't have time to go into today. But instead, what I wanted to show you is the other part of this story, which I think I can tell you now. I was a reviewer in this paper, and there was a reviewer, not me, that asked for this data, and they did it, which was quite interesting, or rather illuminating. So this is secondary dengue, this is primary dengue, and by the four dengue viruses. And you can see that the x-axis is the rate of viremia clearance, and you can see that the greater the decline, the lower the risk. Right? So it's not just how high the viremia level is in the secondary dengue that drives severe disease, but if you can clear the virus fast enough, you lower your risk. Okay? So that then raises this question, well, the dengue infection, if you can control it quickly, you can reduce the risk of severe disease. Then the question is, what controls dengue infection? What clears it? So for a long time, of course, the field is very antibody-centric. Everyone's thinking of neutralizing antibodies. But in the last, about maybe 10 years, and certainly this was my turning point. By the way, I'm not a T-cell biologist, but I'm becoming a slow convert to T-cell because of a colleague like Antonio Bertoletti. So this is a patient with renal transplant. So she's a young lady, about 24 years old when this happened, with a lupus nephritis. So she needed a kidney transplant, and she received a transplant from an unrelated donor. Because of that, she was on a cocktail of immunosuppressive drugs. 3 months later, she caught dengue virus infection. So the x-axis, so this is a summary, the x-axis here, is 0, 5, 10, 15. In a normal person, this will be in days. But the x-axis is actually in months because she could not clear the infection, right? This basically summarized it all, right? So the RNA-emia lasted for about 5 months. We could even isolate virus from the urine for up to 9 months. And the reason why she could not clear the infection is not because of antibodies. She made high titers of neutralizing antibodies against dengue 3, right? For as early as 4 months. And the reason why we have 4 months is because of Paul Tambaya, who is here, who called me one day and said there's something wrong with this case. The lab is making a mistake, but actually the lab did not. It's just that she couldn't clear the virus infection. And we could measure antibody titers, but what she was missing was T cells, and both CD4 and CD8 T cells were low. This is not antigen specific. The dotted line shows you the lower limit of the normal levels of CD8 T cells. And you can see that for a long time, because of the cocktail of drugs, she was immunosuppressed. But because of this worry that she now cannot clear an infection, then the clinicians tailed back some of the drugs. And by 11 months, the CD8 levels climbed up to the lower limit of normal, and she completely cleared the infection at that point. Right? So suggesting that actually it's T cells that's critical for clearing an infection. So the idea that now if you need to drop your viremia quickly, then you might need T cells to do that. And the evidence then started to come from shortly after that, these series of studies. There's a study done in Cambodia. It was a case cluster study. So what the investigators did was once they diagnosed a case of dengue, they went to the household of the child with dengue, and they bled everyone around that lived in the same household, right? And so they could discover those that were viremic but asymptomatic, okay? So then they tracked these people longitudinally for several weeks to know whether they eventually develop symptoms or not, right? And so then you have those that remain asymptomatic throughout the period of observation, those with dengue fever, in yellow, in red is dengue hemorrhagic fever, and then the more severe form, the dengue shock syndrome. And this is the viremia levels, right? And then they asked what differentiated these people, the asymptomatic from those with clinical dengue. So ASD is asymptomatic, CD is clinical dengue. The answer is higher levels of CD4 T cells and higher levels of CD8 T cells. The opposite is true for those with, when you measured antibodies. So those with clinical dengue had higher titers of antibody, right? Suggesting that despite infection and reasonably high levels of viremia, you can be asymptomatic if you mount a T cell response early enough to control the infection. So that all that is with natural infection, but ultimately what we want to do is to be able to vaccinate the population and then protect them against dengue. If not dengue, then severe dengue, then does it hold that cellular immunity is still critical? So just to again orientate you where the T cells are, so these are the structural genes, the code for the capsid, the M membrane and the envelope, which is preformed, and then these are the non-structural genes that have to be expressed inside the cells. And you can see that the T cells target the capsid mostly, and then the non-structural proteins, whereas the structural ones are mostly antibodies, right? So there's unlike the spike protein of SARS-CoV-2, you don't find T cells and B cell epitopes on the same target. And then these are the vaccines, but we'll not go into it today. Maybe you can ask me during Q&A. But what I want to show you is this. This is actually a vaccine effectiveness study. The paper just came out last week or 2 weeks ago. This is a heroic effort, right? So this was during the 2024 outbreak of dengue in Brazil, and the study was done in Sao Paulo. Sao Paulo is southeast of Brazil, right? It had not had a lot of dengue until recent years. So majority of the population in Sao Paulo never had dengue before. And then they had this outbreak. And so the government decided that they would start vaccinating, and Julio Croda and colleagues in Sao Paulo then started this vaccine effectiveness study where they enrolled hundreds of thousands of people into the study in a very short time, okay? So this is a, oops, sorry. Okay. So this is the outbreak. So just to show you the clinically confirmed, clinically diagnosed and lab confirmed dengue. And then, of course, the vaccination, as in most cases, by the time you start, is actually already at the peak of the outbreak, right? But nonetheless, they managed to get the people vaccinated. But the majority, as you can see, only received 1 dose in a 2-dose vaccine, okay? And then at the bottom tells you the different dengue types. They had mainly dengue 1 at the start of the outbreak, then came dengue 2, and then towards the end, dengue 3. Okay, so effectively, we're looking at this part of the events. And there, the vaccine effectiveness, this is a test-negative design, so about 50 to 60%. Now, we can debate the problems or the benefits of a test-negative design later, but this is not true placebo-controlled trial. Okay, but the data that I thought was truly fascinating was this. So this is for time from first dose, right? And the dotted line shows you the vaccine, the mark for vaccine effectiveness. And you can see that for both, whether you're seronegative or seropositive at baseline, or no matter how you, crude or adjusted, the vaccine effectiveness crosses the bar at 8 days. It's very early, right? And why I'm excited at this is because shortly after the mRNA vaccine was, the phase 3 results came out, and talking with Antonio and colleagues, I was fascinated by this line, right? So this is a 2-dose vaccine given, this is a BNT162b2. You see the same trend with Moderna's vaccine as well. This is a 2-dose vaccine given 21 days apart. But you can see that actually the vaccine efficacy did not require a second dose. It only needed a first dose because the 2 lines are separated. So this is the vaccinated group, this is the group that got placebo, and you can see that the authors were aware of this enough to blow it up, and you can see that at 12 days, the lines diverged. Because the incubation period of SARS-CoV-2 infection is about 2 to 5 days, then whatever protected at 12 days must happen about 5 days before. And 5 days before, in day 7, you could only find T cells. There was nothing else. There was no antibodies, there was nothing else, right? And at day 10, you start to find IgM, IgG, IgA by antibodies, but we could not measure any neutralizing antibodies, no matter whether we used LINFAS assay or PRNT. Right? The antibodies could only be detected at day 21. So again, drawing the parallel between the two, I was excited because here's the real-world evidence that probably for dengue, it is the T cells that protect, not the neutralizing antibodies, as we have conventionally thought. Right? So then came this study, which it's a very, very small study. Okay, but I think it speaks volume. So this a vaccine that was co-developed by Walter Reed, the Army Institute in Maryland, and GSK. And so the goal was to develop a live attenuated tetravalent vaccine, but they could not get the viruses to be equally attenuated. So what the Walter Reed, so GSK stopped the program, but what Walter Reed did was to ask, what if we turn this into a prime boost vaccine? So a tetravalent inactivated, so it's the same virus, you inactivate it with formalin and then adjuvant it with alum, and then you boost 6 months later with live attenuated construct, exactly the same viruses, right? And the rationale for this is because that gave them the highest antibody titer. They tried all sorts of combination, live, attenuated first, followed by inactivated, different time intervals and all that, and they settled on this time interval because it gave the highest neutralizing antibody titer. 2 years after vaccination, they then challenged them with a partially attenuated dengue 1 virus. This is the control, so they are not vaccinated. This is the vaccinated group. So 5 out of 6 of them, you can see the viremia started earlier. It's almost like enhancement, right, that we've seen earlier with work of Albert Sabin, right? So on average, say it's about 3 days earlier. So 5 out of 6 of them. 4 out of 5 of these people with breakthrough viremic infection developed abnormal white cell count, abnormal platelet count, and abnormal liver enzymes, suggesting that actually beyond just earlier viremia is actually potentially making disease worse, right? But 5 out of 6. So what happened to the last person? The last person who had no viremia had the highest CD4 count. The person who had viremia but a pristine blood, so no change in white cell count, no change in platelet count, no change in liver enzyme, had the highest CD8 count. And this is antigen specific, right? So again, contradicting the notion that the antibody is critical for protection against dengue virus infection. In fact, these two, I'll hasten to add that these two individuals had they were the two with the lowest neutralizing antibody titer, yet they had protection, whereas those with higher neutralizing antibodies, body titers did not, right? So therefore, that kind of leads us to this last part, which is a collaboration that we had with Antonio, and it was driven by infectious disease physician who was doing a PhD in her lab, to ask the question to what extent T cells can protect against an acute viral infection. We know a lot about chronic viral infections, but what about acute? And the opportunity for us to kind of address that is this. Why is that missing? Okay. So we use 2 vaccines. So these are related to dengue, right? Yellow fever and Japanese encephalitis viruses. So the yellow fever vaccine is arguably the best vaccine that we have in the world, where 1 shot gives you potentially lifelong protection. And it's a full genome, it's a live vaccine, it replicates. 90% of individuals will show viremia. Japanese encephalitis is also a related virus, but in this case, the vaccine makes use of the backbone of yellow fever 17D. We just splice in the PRM and envelop in that and replace those of yellow fever. So essentially, the T cells from one vaccine to another would be identical. It's the antibodies that will differ. Okay, so therefore we then enrolled 33 healthy volunteers. These were tested negative for antibodies at baseline, and then we randomized them into receiving either the JE vaccine first or the yellow fever vaccine, and then about a month later, then they swap over, and this becomes the challenge because both are live and you can measure viremia. And the viremia is shown here that the yellow fever viremia is slightly higher than the Japanese encephalitis chimeric virus viremia, but you can get viremia in the majority of individuals. And the antibody response is highly specific. So if you got the JE vaccine, you only make JE antibodies, not yellow fever. If you got the yellow fever vaccine, you only make yellow fever, but not JE antibodies. But because the T cells are shared, then you can find the T cells across all volunteers. Okay, now what happens at challenge? So in the interest of time, I'm just going to show you the JE vaccinated and the yellow fever challenge, right? And the viremia we will compare across these 2 groups. So I'm going to show you the yellow fever vaccination followed by JE's challenge. And you can see straight away that the viremia levels are lower. This is RNA-emia, not viremia, lower. What's remarkable is that now you have increased number of proportion who tested negative for RNA-emia. Not only did they test negative, they also failed, 3 of them failed to seroconvert. Whereas if you give just the vaccine alone, then 100% seroconversion was observed, right? So if you don't have viremia and you lack seroconversion, by definition, that's sterilizing immunity, which in the past was thought only possible with neutralizing antibodies. But now you're seeing it with T cells, right? To cut a very long story short, so that we ran through the whole thing from antibodies, binding antibodies, innate immune responses, everything. We could not find a correlation except for T cells, and not just T cells, it's T cells against the capsid, where you get this negative correlation with viremia. So the x-axis is T cells measured using the cytokine release assay that Antonio's lab developed for SARS-CoV-2, but we adapted for yellow fever. And this is pre-challenge and this is post-challenge. And again, you see the correlation. You don't see it with T cells from the other parts of the proteome. So why capsid? Because the capsid captures the RNA. And once upon infection, your E protein will fuse with the endosomal membrane. The entire nucleocapsid will then be released into the cytoplasm, but somehow the RNA needs to separate itself from the capsid. Otherwise, it cannot be translated. And the separation requires the capsid to be ubiquitinated. So I think what happens is that the ubiquitinated capsid is then trafficked through for presentation on MHC class 1 and presented to CD8 T cells, which then kills without translation. Right? And that's why if you have enough capsid-specific T cells and you can react early enough, you don't get RNAemia and you don't seroconvert. So let me then try and pull all this together. So what we're thinking is this, right? That we can actually reconstruct the whole thing. This is actually appeared in a commentary to a paper that I will refer to here, but I don't have time to show the data. It's a very nice paper from Tineke Kanter's group in Cambodia. So if this y-axis is viremia, and this is time from infection, so if you have a dengue virus infection and you have high enough pre-existing neutralizing antibodies, then you will block infection, and that's the end of the story. If infection does break through, then you will get T cell response, which if it controls it early enough, you again will not get any RNAemia, or you won't get any seroconversion. Again, the story ends in terms of disease. The problem comes when these two factors wane with time, right? And we know neutralizing antibody wanes, and I believe Tao Dong yesterday also showed CD8 T cells wane with time, at least in the peripheral blood. So then what happens? So when you do get breakthrough infection, then what we know from these papers down here, again, several of them from Cambodia, but not exclusively from Cambodia, has been observed elsewhere as well, that if you mount an early T cell response, in particular a highly differentiated, terminally differentiated CD4 T cell response expressing TEMRA markers, as well as CD8 that express high levels of interferon gamma and cytotoxic granules, then you control the viremia. And when you're able to control the viremia, you either get asymptomatic or subclinical dengue or uncomplicated dengue. Then why is it that some people, what do we need to trigger this level of T cells? And the answer appears to be type 1 interferon. In these papers, again, they didn't attribute it to it because they did measure interferon and they found it high in those with mild dengue but low in those with severe dengue. But there's a very nice review by I forget the first name, Krause et al. In Nature Reviews Immunology, showing how the timing of type 1 interferon is critical, or timing of type 1 interferon relative to antigen presentation to T cells is critical to shape the T cell response into whether it is activated or whether it adopts an exhausted phenotype, or rather express inhibitory markers. Now, then if this doesn't happen, then what then happens is that when you do mount, if type 1 interferon level is low and you delay the activation of these T cells, then when they are activated, they're activated also in the context of this very pro-inflammatory response, which then collectively leads to this dysfunctional T cell response. In this paper by Gonella et al., they showed that it shows that it adopts a TEM phenotype, but others have shown that it is probably dysfunctional, and that they also find a lot of plasmablast expansion at that point. And all this is consistent with what we've learned from COVID-19, where the work of John Werry, where they monitored those who with breakthrough infection after having been vaccinated with mRNA vaccine. The earliest anamnestic response are both CD4 and CD8 T cells. The B cell response does not happen till the second week of illness, right? And I think they're seeing the same kinetics here. It's just that in these kinetics, they're happening in the context of all these pro-inflammatory cytokines that also are probably sending a lot of negative feedback signals into T cells and NK cells, and therefore collectively leading to dysfunctional phenotype. So I hope that gives some thought to how we can now think of dengue. And essentially, I think the message is this, that there are other things that I have not had time to go through, but that we do not, I think we do not need to fear that the vaccine will do this. It's a very different beast entirely from wild-type dengue virus infection, and that if with this understanding of pathogenesis, we should stop saying that dengue pathogenesis is unknown. That we do know it. So with that, I'll leave it, and thank you very much. Happy to take questions. Hi.
Thank you for the presentation. So for the T-cell response of the COVID-19 patient, I have read some papers, and also from the my group, the level of the T-cells are positively related with the severity. But for the dengue, it is negative correlated. Yeah. So do you think it is the sampling time different, yep, or the index different?
Yeah, I agree. I think it's sampling time. That's one issue. Yeah. I think it's also the virus, so and the host, right? So what I didn't show you is that the dengue virus itself is very different. So although there are just 4 dengue viruses in a phylogenetic tree, within each dengue type, there's quite a diverse range of genotypes and even different strains of dengue virus. And each of them, even as subtle as one single amino acid substitution, can make a profound change to how the cell would react to the infection. And that reaction is, a lot of it is type 1 interferon response. And so if the type 1 interferon response happens too early before antigen presentation, then even if you have T cells, it's going to go into this inhibitory kind of phenotype, which then impairs clearance. And therefore, when you just measure T cell, it looks like it's driving disease, severe disease. But actually, the event that's critical happened before that, right? That's one. But second one is also the host. So we know that in older adults, we have auto antibodies against type 1 interferon. And so in those individuals, even if they do express type 1 interferon, the interferon cannot act on interferon alpha receptors to trigger the necessary response, right? So I think that's why I think we can't look at a time once illness onset happens. It's really difficult because there's a lot of noise. Mm-hmm. So we have to go slightly before that, and there the power of cohort studies is where we probably can glean the most information.
Okay, thank you.
Thank you.
You showed that in the vaccinated cohort that then got infected, the viremia came earlier, right?
Yeah.
But there was no difference in viremia in the first versus second infection.
Yes.
So you think it's just a question of timing, but even in the vaccinated, the level is the same, or?
Yes, that's right. I think it's a difference in timing. Many years ago when dengue first started, Albert Sabin did a human challenge because there was no other way of measuring dengue virus. So he literally took blood from a dengue patient and gave it to someone who was healthy. He saw the same thing. The fever started earlier when you give to someone who has had dengue before, right? So I think that it tells you that the process is probably more efficient, but it's not changing the overall dengue burden. But somehow the host response is then able to drive disease. I should add that actually inactivated dengue virus prime and then live attenuated boost had a second part to the story. The second part of the story was this: despite lower neutralizing antibody titer with just the live attenuated vaccine alone, 1 dose, actually when they did the same thing, there was 100% protection.
Thank you for the wonderful talk. A question about how confident that if you want to recommend to design a dengue vaccine to go including capsid?
Okay, great question, and good to see you, Ken. I think so. The problem with capsid is it's very small, right? So I'm not sure that 100% of people will react to the capsid by making T cells against it because of the diversity of the HLA. But that capsid is important only to, in our study, we used the viremia as an endpoint, right? But in reality, if you have, what we yet have gotten to show or gotten to evaluate is whether T cells against the other parts of the virus can stop the development of severe disease. So lead to a more rapid clearance of dengue viremia and therefore reduce the risk of severe disease. Because if that's possible, that is helpful as well. That is beneficial to the population.
Okay, so how about the, I look at your yellow fever challenge study.
Yeah.
Probably the sample size limited, And when I look at the slide that the day 4 after the challenge, it looked like not only capsid, NS3 is very close to the p-value below
Possibly.
0.05.
Yeah, you're right. The population size is small, and so there potentially that's why we could see only capsid, because the rest, and the biology-wise, the rest needs to be translated. So it's a slightly later event than the T-cell reaction against capsid.
So how about NS1 to be included?
Yeah, I mean, I think to me, the pathogenic role of NS1, to me, is still debatable. I mean, for those in the audience, NS1, besides forming the replication complex, is also secreted, and something that the secreted form of NS1 then acts like a toxin to bind like receptors like TLR4 and trigger inflammation. I'm not, clinically, I think it's very difficult to support that hypothesis because patients with primary dengue have high levels of NS1. And the NS1 can last, 50% of them, 3 weeks after they recover from dengue, they are still NS1 positive. If that were a toxin, they would be dead by then, right? So I'm not a fan of NS1 as a pathogenic factor, based purely from a clinical point of view. But I think for a vaccine, though, there's no harm putting it in, right? Because antibodies against NS1 can trigger complement and all that can also lead to more rapid clearance of the virus.
Thank you very much.
Thanks.